The purpose of this experiment is to find how many milliliters of water flow out of one stoma in a mint plant per hour. To achieve this, the transpiration rate of the whole plant will be calculated by taking the initial mass of a mint plant, then wrapping it so that no water is able to evaporate through the soil, and then taking its mass over the course of one day. Then, the total number of stomata on the plant will be calculated by counting how many stomata the mint plant in a specific area has under the microscope. This value of stomata counted in the area will then be used as a conversion factor along with the mass of the mint leaf with an area of 1 centimeter squared will then be used to estimate how many stomata are present on the mint plant. With this data collected, the rate of transpiration for the whole plant can be divided by the total number of stomata on the whole plant in order to get the rate of transpiration for one stoma on the plant.
After conducting the experiment, the rate of transpiration per stoma is found to be about 1 times 10 to the negative 7 milliliters per hour.
For the procedure in determining the transpiration rate, we must first make sure that the plant being used in the experiment is watered well. Then we must take the plant out of its plastic container and wrap it in saran wrap so that the soil is completely encompassed and covered. This must be covered to the point that only the plant stems and the leaves should be protruding from the saran wrap. Then, take the initial mass of the plant that is wrapped in the saran wrap and record it in the data table. Then after that, you just take the mass of the plant periodically over the next day and record the date and time that the mass was taken, and also record the mass of the plant. To determine the number of stomata on the plant, you must first take clear nail polish and apply it to a 1 centimeter squared section of a selected plant leaf. Then, allow the nail polish to dry thoroughly for about 10 to 25 minutes. Once the nail polish is dried, take a piece of clear cellophane tape and place it over the area where the nail polish was applied on the leaf. Slowly take the piece of tape off of the leaf, making sure that the nail polish peel is attached to the tape. Then apply this piece of tape to a clean microscope slide. Then examine the slide with a microscope under a 40 times magnified objective lens. Count how many stomata are present in the field of view and record the number in the data table. Then take the mass of the section of the leaf that has an area of 1 centimeter squared and record it in the data table. Then take the mass of all of the plant leaves and record it in the data table. Lastly, using the number of stomata per area of field of view in centimeters squared and the mass of the leaf per centimeter squared as conversion factors, calculate the total number of stomata on the plant.
The calculations followed the graphing and allowed us to determine the results of the lab. The first step of this process was to determine the field of view of the 40 times objective that we used to look at the stomata. The diameter of a 40 times objective is 0.4 millimeters, making the area of the field of view 0.12566 millimeters squared. We counted 38 stomata in this field of view, which let us extrapolate this to 30,204.3 stomata per centimeter squared. Next, we had to determine the total surface area of the leaves of the plant, and we chose the massing method. We determined that 1 centimeter squared of mint leaf weighed 0.012 grams per centimeter squared. With a total of 10.783 grams of leaves on one plant, this was approximately 898.583 square centimeters of leaves in total. The product of these two figures results in about 27,173,430 stomata on the whole plant. Finally, we had to take the slope of the line of best fit from the graph, which was the amount of milliliters per hour that the whole plant lost, and divide it by the total number of stomata that the plant contained. After adjusting for significant figures, Our final result was that the average mint stomata lost 1/10 millionth of 1 milliliter of water per hour. After collecting and plotting the data on a graph, the slope of the line of best fit derived from the graph, defined as the change in the y variable over the change in the x variable, was analyzed. In the experiment, the independent/x variable was the time elapsed after the plant was wrapped in saran wrap, and the dependent/y variable was the mass of the plant at the specific time. The experimental design caused for the only source of the mass change in the plant was water leaving the mint plant through its leaves. Because the density of water is 1 gram per milliliter, it can also be said that as the plant loses 1 gram of water, it loses a milliliter of water as well. Because of this, the slope of the line of best fit represents the loss of water of the whole plant in milliliters over time elapsed in hours, which is the rate of transpiration for the whole plant. To calculate the total number of stomata, which are the pores on the leaf surface that allow for substances to move in and out of the plant, multiple conversion factors were used. As shown through the calculations, the number of stomata present per centimeter squared, the mass of the mint leaf per centimeter squared, and the total mass of the mint plant leaves were known measured values. With these measured values, dimensional analysis can be performed by converting the total mass of the leaves into area and using the total area of the leaves to estimate the total number of stomata on the plant leaves. With these known values, the rate of transpiration per 1 stoma can be calculated by dividing the rate of transpiration of the whole plant by the total estimated number of stomata on the plant, yielding the value of approximately 1 times 10 to the negative 7 milliliters per hour per stoma for the mint plant.